Grade 9, Lesson 5, the fundamental unit of life.
In this video, I will discuss about osmosis. But before telling you about that, I should explain you the three terms solute, solvent and solution.
Now let us understand how a solution is made. When a solute is dissolved in a solvent, a solution is made. Now what is a solute? A substance. For example, if we take sugar or salt and mix it in a solvent, for example water, we get a solution that is either a salt solution or a sugar solution. So here the solute is salt or sugar and solvent is water. Solvent can be any other liquid also apart from water. So if we dissolve solute in solvent, we get a solution. While studying the effects of different solutions on plant and animal cells, three conditions may arise depending upon the tonicity of the solution. Now, the term tonicity can be defined as relative solute concentrations of two environments separated by a semi-permeable membrane. In other words, by comparing the tonicity of the solution, we can determine the direction in which osmosis will occur. Now let us discuss the three types of solutions, that is isotonic solution, hypertonic solution and hypotonic solution. So now isotonic. Iso means same. Tonic means strength. So same concentration of solute and solvent is present in the solution, then it is known as isotonic solution. Hypertonic. Hyper means more. Tonic means strength. So more solute and less solvent concentration is there in the solution, then that solution is known as hypertonic solution. Now hypotonic solution. Hypo means less. Tonic means strength. So when fewer solutes and more solvent concentration is there in the solution, then that solution is known as hypotonic solution. Let us discuss some real-life examples related to the three types of solutions. The first is example of the hypertonic solution. So when you put pasta in a pot of water, when you put in the pasta, since it has little water, absorbs the boiling water you put in and making it grow. So this was the example of a hypertonic solution. Another is hypertonic. So example for hypertonic is when you put your clothes on a clothesline to dry them off, since the air is hypertonic to the wet clothes, the water comes out and evaporates. Another example is if you stay in the shower too long, you will notice that your skin can gain water, and this is often called pruning. This was the example of hypertonic solution. Now isotonic. Example for isotonic is when doctor gives you blood of your own type through a syringe. So the cells have the same kind of molecules as the cells already inside the stream. So the new cells aren't hurt by the ones you already had. Another example, if you clean contact lenses, you use an isotonic saline solution to clean the protein from your lenses. What happens when you put a plant cell or an animal cell in hypertonic, hypotonic and isotonic solutions? Hypertonic solution. A solution that is dilute or has more water molecules than what is present in a cell is said to be hypotonic. If an animal cell is placed in a hypotonic solution, water from the solution outside moves into the cell by virtue of osmosis. As a result, the cell swells up. If the process continues, the cell may burst. So please note that water molecules will move across the membrane on both sides. However, there will be a greater movement of water molecules into the cell than outside. That is, the net movement of water is inwards. Similarly, If a plant cell is placed in a hypertonic solution, it does not burst. A plant cell is surrounded by a tough cell wall, hence the cell becomes turgid when water enters it, but it does not burst. Isotonic solution. If the concentration of water molecules in the solution is the same as that of the cell, there will be no net movement of water molecules across the membrane. So water molecules can move from an external medium into the cell and vice versa. However, the net movement of water molecules is zero. Such a solution is said to be isotonic with the cell. For example, if there is 10% NaCl solution outside the cell and 10% NaCl solution inside the cell, then there will be no net movement of the molecules in and out of the cell. So that type of solution is known as isotonic solution. Hypertonic solution. In case the solution has less water molecules than that of the cell, it is said to be a hypertonic solution, or a solution that has more solutes and less water in comparison with the cell. That type of solution is known as hypertonic solution. For example, now if an animal cell is placed in a hypertonic solution, it loses water by osmosis and shrinks. Similarly, a plant cell, however, does not shrink when placed in a hypertonic solution. Why? Because a rigid cell wall prevents its shrinkage. However, as water or cell sap moves out of the cell, the protoplasm starts shrinking. In other words, the cell membrane along with the cytoplasmic contents recedes away from the cell wall, and this process is also known as plasmolysis. So let us talk about the types of osmosis: endoosmosis and exoosmosis. First is endoosmosis. So endo means inside, in, that is the inflow of solvent or water into a cell from outside is known as endoosmosis. So when a cell is placed in hypertonic or dilute solution, the cell swells up in this case. Another is exoosmosis. Exo means exit or outside or outward. That is the outward flow of water from the cell when placed in more concentrated solution is called exoosmosis. For example, solution like sugar solution. So when a cell is placed in a sugar solution or maybe a salt solution, then the cell shrinks in this case due to exoosmosis. Example of endoosmosis. If we take some raisins and put it in distilled water overnight, so we will see after few hours the raisins swell up. So the process involved in this case is endoosmosis. That is, when the cell is placed in distilled water or a dilute solution, the water from outside the cell enters inside the cell and the cell swells up as a result of endoosmosis. Example of exoosmosis. So, if we take some grapes and put them in sugar solution overnight or for a few hours, so after a few hours you will observe that the grapes shrink. So the process involved in this case is exoosmosis, that is the outward flow of water from the cell when placed in more concentrated solution. So the water formed inside the grapes come outside in the solution, and as a result the grapes shrink due to exoosmosis.